I'm going to talk about the etiopathogenesis of the nephrotic syndrome. First, the nephrotic syndrome. What do you mean by nephrotic syndrome? It is not a single disease. It is not a disease. It is a syndrome. Means the clinical features or the symptoms or the clinical manifestations that results from various type of the kidney disease, that is glomerulonephritis. It may be either primary or it may be either secondary. So I'm going to talk about the primary causes. So before going to this, what is the cardinal features of the nephrotic syndrome? There is heavy or massive proteinuria, that is more than 3.5 gram per day, hyperlipidemia, generalized edema, that is called anasarca, and the hypoalbuminemia, that is the plasma level of albumin is less than 3 gram per dl. Okay, so the primary causes, as you see, this is the minimal change disease, which is most common in case of children, membranous glomerulonephritis in case of adult, membranoproliferative glomerulonephritis, focal segmental glomerulosclerosis. The pathogenesis of the minimal change disease. It is cell mediated, means there is the activation of CD4 and the CD8 type of the cells. And in case of MGN, in situ means what? The antigen antibody reactions occurs within the kidney, as the glomeruli contain three structures. One is the subendothelial region, second basement membrane, and third one is the epithelial region that contain the podocytes. Okay, so glycoprotein 330, it present within the sub means the basement membrane layer, in between the basement membrane and the epithelial layer. In some case of autoimmune disease, this glycoprotein 330 act as a antigen which is fixed against it the antibody produced, and where the antigen antibody reactions occurs and that immune complex get deposited in this region. In MPGN, FSGN and FSGS, all are immune complex mediated grossly. In case of MCD, the kidney look like normal, but in these four, the kidney get enlarged. The light microscopy. Light microscopy means what? What we see in case of light microscopy, either there is any change occurs in glomeruli or not. That should be seen. So in case of minimal change disease, as you see, there is no any changes seen, but slightly in some cases there is the proliferation of the mesangial cells only, and in case of tubules, renal tubules, there is the deposition of the lipid. That's why it is also called lipoid nephropathy. Now in case of MGN and MPGN, as you see, this one is the Bowman's corpuscles, this one is the glomeruli, and this is the Bowman's space. In case of Bowman glomerular, these are the capillaries. Along these capillaries, there is the basement membrane that gets thickened and form a spike-like structures. But there is no any proliferation of the mesangial cell or the inflammatory cells. But in case of MPGN, as you see, there is the thickening of the basement membrane, but along the thickening, there is the proliferation of the mesangial cell and also the inflammatory cells, that is called neutrophils. Okay, so in case of MGN, there is only the thickening, but in case of MPGN, there is the thickening plus proliferation of the mesangial cell and the inflammatory cells. Now in FSGN and FSGS, first the focal segmental glomerulonephritis. Focal means what? As there are many glomeruli. From these glomeruli, one glomeruli get affected. And segmental means what? Half part of one glomeruli get affected. As you see, in case of focal segmental glomerulonephritis, this glomeruli is normal, but in this case, half part is normal, but half part get proliferate, mesangial cell proliferation. But in case of FSGS, this glomeruli is normal, but in this case, half part is normal, but in case half part, there is the proliferation, but in this case, this get sclerosed or undergoes the hyalinosis. Okay, so now electron microscopy. So electron microscopy is done to know the exact location of the immune complex deposition. In case of MCD, there is no any deposition of the immune complex, as it is the cell mediated type of the disease. But in this case, there is the flattening of the podocytes, and the podocytes get damaged, and they get interconnected to each other and get flattened. That is called effervescence, and this is the hallmark for the MCD to diagnose this case. And in case of MPGN, as you see, there is the thickening of the basement membrane and it form a tram track appearance. That means the true parallel line, like. Okay, so now in case of FSGS, as there is the deposition of the immune complex in the mesangial layer, not in the subepithelial or subendothelial or in the basement membrane. It get deposited around the mesangial layer. Okay, so now the immunofluorescence. Immunofluorescence means what type of immune complex get deposited. Either this one is the IgG or C3 or IgA or IgM. In case of MCD, there is no deposition, but in case of MGN, there is a granular deposition. In case of MPGN, same as MGN, as you see in this figure, this is the granular type deposition. Okay, so now the pathogenesis of the nephrotic syndrome, as it is caused both by the minimal and the membranous. In case of minimal, it is cell mediated type, but in case of membranous, it is antibody mediated type. So I'm going with the antibody mediated type. As from the blood the antibody enters into the kidney to the renal artery, then it enters into the capillaries, glomerular capillaries, then finally it enters into the subepithelial layer. Now the antigen present into the subepithelial layer get activated, and these both get reacted to form a immune complex. Due to this, there is the alteration in the membrane, the glomerular layer get injured, and what happens, there is the alteration in the negative charge. That means the negative charge get neutralized, and there is increase in the size of the pore. Through this pore, large amount of the proteins get excreted out, and finally these proteins get absorbed into the tubular cells, and that get catabolized in the kidney, and that results into the increase in the protein loss from the body. To compensate it, the liver synthesizes large amount of the plasma proteins. Along this plasma protein, lipoproteins are also synthesized, that is LDL, VLDL, TG, triglycerides and the cholesterols. Due to increase in the synthesis of the lipoproteins, the concentration of lipid increase in the blood, and that leads to hyperlipidemia. Due to hyperlipidemia, large amount of the lipid get excreted, and that leads to lipiduria. That means the escape of lipid through the damaged GBM. When the liver cannot compensate the protein loss anymore, then what happens? There is the hypoalbuminemia. Due to hypoalbuminemia, as albumin is responsible for maintaining the osmotic pressure, and there is the least amount of the albumin, then what happens? There is the decrease in the oncotic pressure. Due to decrease in the oncotic pressure, now the fluids from the vessels moves into the extracellular space, and that is the extravasation of the fluid, and that leads to hypovolemia. Due to hypovolemia, the aldosterone and the areas concentration increase, and that further causes sodium and water retentions, and it causes more extravasation of the fluid, and finally it leads to anasarca, that means the generalized edema, and finally the kidney look like this. As you see, there is the thrombosis due to the increase in the number of fibrinogen or decrease in the anticoagulant as the proteins get excreted through the kidney. Due to this also the thrombosis happens. Now this is the pathogenesis of